let me go forward anyway and say, hi, my name is Martin Peacock. Today we're doing a webinar titled Data Science for Biosensor Designers. I want to be quick and efficient with your time today. Feel free to put things into the chat and the Q&A buttons. We will try and monitor them. So in the first instance, I'll probably be mostly talking. So I won't necessarily be able to monitor the chat and the Q&As immediately, but we will try and be responsive. So this is the Data Science for Biosensor Developers webinar. If AFTAB lets people into the room as they turn up, 9th of October. We appreciate that. This is primarily for our colleagues in India, and it's been organised by Technando, who are our distributors and partners actually in India. And so we thank them for that, and we also appreciate that you guys have come at sort of fairly late in the evening, so it is appreciated. I'll go forward a little bit. I'll try to be quick with you because I want to be respectful. So I'll do a quick introduction. We'll talk. This is about data science for biosensor developers, but we will talk about different types of data, and it will be in a specific domain, which will be electrochemical sensing and electrochemical assays and electrochemical biosensors. But we will talk about how we deal with cyclovoltammetry. We will talk about how we deal with amperometry. We will talk about how we use data science in our manufacturing, and we'll also talk about how we use sort of data science in our impedance data. Then we'll also talk about how we apply data science. Well, what does data science do for ZP? It really means that we can actually go from idea to product really quickly. And when I started my career, I would say, and I don't want to say 23 years ago, but when I did start that career after my PhD, data science and the use of data science was just not so strong. So we would have to make really good sensors in order to kind of have any kind of clinical usefulness. But there's so many good tools out there these days, and the sort of understanding of data science is so much stronger that actually we don't have to spend years trying to make the world's best sensor. We can mix a good sensor with good maths, and we end up with a good system. So we will demonstrate some of the practical implications of what comes out of this kind of combination of science and data science. My sort of definition of data science, I realize now that in retrospect, anyone who's kind of got a science and engineering background, you already are partially a data scientist. I've always done statistics, simple linear regression. It's just now we sort of formally recognize this is what we're doing. And once you recognize this is what I'm doing, it really helps you then, because you can sort of search around, watch those kind of YouTube videos, come up to speed. So data science for me is the sort of application of the scientific method. We have processes which help us in data science at ZP, and I will touch upon that. We kind of exploit data science through algorithms, and then we use it to really, data science is not a subject for its own sake. It's actually a subject for extracting information and actionable information out of data. And I say here about structured and unstructured. We'll mostly today be talking about structured data sets, and you'll see that our processes mean that we actually end up getting quite structured data sets. So just understand that it's not just pool or lakes of data that we'll be talking about today. We will be talking about how we have processes starting in our lab, which end up with data kind of in quite a structured way. As I say, for us at ZP, we love science, we love engineering, but really we want to see its sort of commercialization. So data science is really important for us because it actually allows us to make business decisions. And for the simplest application of data science, like in our business, is KPIs, key performance indicators, to show that our actual manufacturing is operating correctly. So data science is a combination of statistics. It doesn't always have to be complex statistics. It could be mathematics. It doesn't always have to be complex mathematics. A lot of what makes data science much more powerful these days is the programming automation, and we will talk about that today. That if we come up with a code that will actually help us analyze our data in some way, then we want to automate it so that the rest of the company can essentially benefit from it. And also, data science, we have a certain domain knowledge, which is, it happens to be electrochemical and chemical, and we often apply it into food quality, food safety, precision agriculture, and health applications as well. I'm not going to spend a lot of time saying, talking about ZP. We've been around for 9 years now. We are ISO 13485. We do a lot of development. We primarily do it in the UK and Norway. We have an international team, and our facilities really are kind of state of the art. And as I say, we've got UK and Norway is primarily where we're at. We do have a data management system called Julie. So a lot of the data science, we've automated it by actually coding it into Julie. And you can get access to Julie. If you're accessing Julie and you've got feedback for us, just give us the feedback, because we are interested in people sort of piling into the Julie system, using it, giving us feedback. Feedback can also be critique, and we're happy to receive that. So data science is at ZP is mostly expressed through Julie. I talked about, at the beginning of it, that data science can involve structured and unstructured data. For ZP, we end up structuring our data. If you're a biosensor developer, it's really important that from the day, almost day zero, day one, that you are actually coming up with some way of structuring your data. So many companies and so many R&D efforts, by the time they get to year 3, they've forgotten what they did in month 1. But at ZP, we've always had this kind of data storage structured, so we can go back almost a decade now to find old data. So Julie for us is about data storage. So we have structured data, which makes the data science easy. The first part of data science, I'd actually say, is visualization. So Julie allows us to essentially have a string of numbers. So it could be current versus time, or it could be current versus potential, and we can visualize it. Human brain is really good at pattern recognition. So, we do like to visualize our data pretty quickly. We can process our data. Processing could be turning raw signal into information. I think that's really the kind of one of the best definitions of data science. Julie for us is also about data acquisition. You'll see that happening in real time. And just as an FYI, if Julie's a sort of data lake, then you have to be able to pull data out of Julie, and that's why we have an API, an application program interface, as well. I'll go. The one thing about data science and the way we use data science is that at any one time, there'll be more than one person at ZP kind of looking at the data. So I'll give you a scenario that we're having a meeting last night with somebody who's using some of our technology in India. So they're in India, they're in a lab in India, they're gathering data and it's going up to the cloud, at which point then I'm looking at the data. One of our engineers, Solron's looking at the data. One of our engineers called Sindra has looked at the data. Aftab, who's online with us today, has looked at the data. And then one of our colleagues called Niti has also looked at the data. So data science becomes very powerful when you can essentially upload it, visualize it, and other people can all look at the data together, because that's really a network effect. That one person looking at the data is one thing, but once you've got 2 people or 3 people or 4 people looking at the data, it just becomes, everyone gives it an insight. So data sharing and data visualization is really important, and that's what sort of Julie does for us. This is not a Julie webinar, but I am just going to say, very quickly, that you can sign up for a free account. And I appreciate lots of you did sign up for a free account, because you give us feedback. You can give us feedback on its utilization, and you can also give us a feedback on its costs, and so we can work out the business model properly. So I have mentioned it, but one of the first things in data science asking the question is my data structured or unstructured? So the first thing is that if we're in our lab developing a biosensor, then we have a process where everyone who's in the lab, if they're gathering voltammetry data, is uploading it into a database. So it's becoming structural already. So we are structuring our data kind of in real time, and that is a process, which is, it's also a culture. Process and culture are kind of, you can't have a process unless you have a culture of people actually doing it. So we do have a process, and then we kind of get everyone to do it, and eventually it becomes the company culture that all our data ends up structured in our Julie database. And then it means that we can see it all online, we can visualize it, and we can all start processing it. A typical biosensor workflow for us at ZP is, now this image is obviously of a screen printed electrode. So maybe this conversation is more specific about IVD, in vitro diagnostics. So somebody comes to us and says, I want to make a sensor for a particular hormone, or I want to make a sensor for a cardiovascular marker. I mean, the classic one is glucose. I'm not, and I understand that glucose is very old, so I'm not going to necessarily use glucose as an example. But somebody's come to us and said, right, first of all, I want to make a in vitro diagnostic sensor. So this, they've commissioned us, so we go into the lab and we take our screen printed electrodes with us, and we will functionalize it. Now, we've tried to simplify the functionalization now because actually we made the electrodes circular, so it's kind of quite easy to either hand pipette onto it or to use a robot to do it. But when you're doing small scale manufacturing or small scale fabrication, what I mean by that, maybe 300 sensors, if you've got the skill, it's just as easy to pipette. And the fact that we've now made it circular, I think actually aids the functionalization. So we'll functionalize it, and we won't just do one and test one. We will try to do 100, if not more. And so this is a data set, for example, where we were doing impedance. And I could see from the image that we've made up like 90 samples. So our workflow, I mean, the title of this webinar is Data Science for Biosensor Developers. So if it's an in vitro diagnostic, we'll often start with the screen printed electrode. We're obviously biased, we're using our own. Because of the circular electrode, it's easy to fabricate. And then we can, if it's a simple one-step process, then it's just pipette, pipette, pipette. If it's two-step process, then it'll be pipette, dry, then pipette, then dry. So, it becomes longer to do that. But we aren't gathering small sets of data. We're actually gathering quite large sets of data. And I showed it here just as a sort of simple linear regression that we'll have a signal versus concentration. But in reality, what we're really doing is kind of really trying to get an idea of the spread of signal within our for each concentration. So here I'm showing it as a sort of box and whisker plot. We really quite like this. I mean, this has really come from our engineers. It's a sort of, the engineers see the value in this. And, for us, this is actually some data that's kind of come out of our lab. Now, what you can say about this is it's not perfect. And I think that's what I really like about data science, actually. You don't have to be perfect these days. You can make more and more sophisticated algorithms in order to deal with non-perfect data. But if I was to just kind of wind up a bit, we go, into the lab, we functionalize these electrodes. We've done more than one, we try to do quite large numbers of them. And then we're plotting the kind of signal out of these things as a function of concentration. And we'll often look at it in a sort of box and whisker plot. And when I look at that, the first thing is the median tells us that, yeah, we do have a signal that's proportional to concentration. So that's a tick for us. And then if I look at it, if you put a purely academic glasses on this, you could actually argue, oh, this is not good enough. I need to have better, I can't have this overlap between the signal from these different concentrations. But I think in reality, there is a philosophy that, what is good enough. And sometimes in a medical diagnostic, you don't need to be 90% accurate. It's nice to be 80% accurate, but actually a diagnostic could be useful at 70% accuracy. And if you're really talking about commercialization, I would suggest that you have sort of milestones that say, in my first iteration, I'm going to be 70%, then I'm going to be 80%, then I'm going to be 90%. So accuracy is a journey. So if this was our first effort, and this is the kind of, sort of thing that we'll get from a first effort, where actually we do have a signal versus concentration, but the spread in data, shows us that it's not perfect, and we could end up, having false positives and false negatives. But in general, I kind of quite sort of happy with it. And so our philosophy, which is really borrowed from MIT, Massachusetts Institute of Technology, is kind of fail fast. So if in the lab you're getting kind of data that you can see your trend, you've gathered it with enough data behind each data point that it's statistically useful, then we would actually be translating it immediately onto a hardware platform. And this is called Sensor. But we would immediately be taking the lab protocol and the sensors that were developed in the lab, putting it on this hardware, and actually going into, I would say, preclinical trials. Literally just going in there, going into some sort of semi-clinical setting and just testing it. Now, it's not saying that we're going into the clinic and then making clinical decisions on it. We're just gathering data. So we do have, be careful about understanding what's good enough in order to move forward, because you can spend years on the left-hand side of this slide trying to make things really perfect with no overlap between these different concentrations. And then you go into the clinic and find out that those years of effort were either just wasted because it doesn't work so well in the clinic, or in fact you over perfected it and you really didn't ever need to be that good. So fail fast or fail quickly, but be successful, but don't spend years, let's say, developing this kind of tech. So I'll give you an example here. That let's say there was a clinic, there was a disease, and there was a threshold. Whether you have or don't have the disease is based on a threshold. Well, when I look at this data here, and if I draw a sort of arbitrary threshold of about 78 here, anything below that line, I'm going to classify as somebody has the disease, and anything above the line, I'm going to classify them as healthy. What you find is when you go into clinical settings that most healthy people are kind of up here. So naturally, most people are not near the threshold. So your ability for a healthy person to otherwise give you a false positive is pretty unlikely. Whereas most, I'm making a scenario here, whereas most sick people, maybe they're actually quite a long way, quite far away from the healthy population. And so even though my data is kind of overlapping and there's a sort of grayness, you will find that it can clinically work. And I'm sort of going out on a limb a little bit. But if I had data like this and I went into a clinical setting, I might find that my accuracy is something like 75% to 80%, which is good enough for the clinic. Especially when it's a sort of binary, does this person have a disease or not have a disease? So be careful with over worrying about your data. You have to be convinced that you have a signal versus concentration, but you can improve accuracy with time. So at ZP, we're really quite good at speed of development. And what I mean by that is we can go into a lab, we can sort of make up a series of concentrations, we can test those concentrations, we have domain knowledge, so we know how to essentially pull the signal out of our data. This is voltammetry. I'm sure many of you are familiar with voltammetry. I recognize lots of the names, including a couple of doctors and professors here, so I'm not going to overplay this, but we are pretty quick at development. And it really comes about because of this kind of rubbish in, rubbish out. So unfortunately, I think what's happening a lot is we want to be quick in our development. And when you look at my workflow starts with a screen printed electrodes, which we functionalize, which we then test, and then we do essentially statistics on it. We do maths on it, linear regression, we do statistics on it, what's our median, what's our range, what's our outliers. We do that kind of analysis. But it all starts with the screen printed electrodes. So at ZP, we're biased. We have a whole series of screen printed electrodes, but we've really worked over the years on actually making sure that these screen printed electrodes are going to give us accurate assays, and I'll sort of touch upon that a little bit. Now, I should mention that this webinar is actually being recorded, so there's no need to necessarily take notes. I'll send you a personal email at the end anyway with a link to the recording on this. But I'm just saying that rubbish in, rubbish out. So what we're trying to say is actually, I think what's holding up a lot of biosensor developers is actually the repeatability of the underlying electrodes. I hear things that I've got a screen printed electrode, and for the same concentration I test it and I have a signal that's double on one electrode versus another electrode. So something at ZP, we absolutely love data science, and we absolutely do. We what we do is we try to control manufacturing, and then we also control our testing, and then we really do check. I mean, it's simple statistics, but we check the mean. Are these electrodes giving us a mean height? What's the standard deviation? But then turn it into relative standard deviation, because it's easier to have a conversation with somebody and say, oh, well, my distribution is about 7%. Oh, okay, I understand 7%. Whereas if you try to say, my standard deviation is 0.314 versus a mean of 4.703, that's it's not so fluent in people's minds. But we often try to get a standard deviation. We always aim for less than 10%. A bit of a rule of thumb is that if you can manufacture something in the diagnostic space with a standard deviation of less than 10%, then you're starting to get to be useful, let's say. So then we look at the median, we look at the min, we look at the max. So, we're looking at the ranges, we're looking at the medians, we're looking at the mean. Obviously, by looking at the mean and the median, we can understand whether our data is normally distributed. So when you look at our number here, I can see it's 4.7 versus 4.67. So you can see that our kind of errors are essentially normally distributed. There's not a sort of, they're not skewed too much. We do this quite a lot. We do a lot of what we call wafer mapping. So we'll take a sheet. So this is sheet one. First of all, I'm going to show you some bad data, because when I look at sheet one and we've done some data on it, I can see that two of the sensors that got tested on sheet one are no good. We'll never ship you sheet one, just out of interest, because sheet one is the first time you print it, you essentially, everything is fresh, everything's just been set up. So we'll dispose of sheet one. It's, but then sheet two is tested. We won't test every sheet, but now we test one of the sheets sort of not at the beginning, but near the beginning. Sheet 6, sheet 18, which may be in the middle of it, sheet 21, which is later on. So all the time we are testing a certain percentage of the population. Now, this is a data science webinar, so I want to give you some insight. So we'll generally test about 5% of the population of sensors, of the screen printed electrodes rather, to essentially pass the batch. So just as a sort of get an understanding of, how much testing is actually going on here. Now, I did say that data science is, for me, becomes most interesting when you can sort of visualize it. So here we will have a table of results. These are different sheets within the same run, but then we can visualize it by actually seeing how the mean signal in those sheets is changing during the run. And it's kind of nice in some ways, because we don't have a constant drift up or constant drift down. You could criticize this, but really, I mean, this is like 5.8, this is 6, so, and then this is 5.8 again, and then this is, approximately kind of getting close to sort of 5.9-ish. So I'm just saying to you that the visualization really helps us, and to see if we've got a drift across our manufacturing run, but we don't necessarily have a drift across our manufacturing run. I do have a few colleagues online today, by the way, so it won't just be me, and I will be a little bit quicker and a bit more respectful to let my colleagues come online as well. But we do analyze our data quite heavily. And I did say that, data science becomes powerful then when you can automate it. So we do automate it. So we will ask, and this is what I like about the questions that we're asking are, what's our peak-to-peak separation? If it's within a certain criteria, then we will pass it. What's our peak-to-peak height criteria? If it's within a certain criteria, we'll test against that criteria, and then we'll pass it. That's that automation part. So, data science, I think, has always been happening in science, but now many more engineers and scientists are actually also programming, and that's what we do. Once we come up with a rule, we can scale ourselves by actually coding that. And you can just see there, that's not that hard. What we're doing is not conceptually that difficult. Something that we'll talk about in a little bit as well is that I don't want to make this the glucose webinar, because I feel like glucose is a very commoditized technology. But Lynn will show you that actually we, just simple linear regression, that even we make glucose sensors, we test glucose sensors, but in testing them, we have a process. We structure our data. So we're ISO 13485, which means we have standard operating procedures. So, somebody's got an instruction, they go into the lab, they put the sensor into a test cell, they start the experiment, they then add some glucose and the signal goes up. And you'll notice that what they're doing is they're actually being told by the SOP, wait for a certain amount of time, add glucose, wait for a certain amount of time, add glucose, wait for a certain amount of time, add glucose. So you can see that, I think that's sometimes the difference between, let's say, industry and academia, that we do have all these processes because we want to be sort of boring, but we need them because then the data comes out in a very consistent manner. And then it means that people like Lynn and Sindra can actually just automate the analysis of that data. So in kind of order of things in a minute, the first person that's going to speak will be Sindra, and Sindra will probably be speaking about voltammetry. There'll be a bit of Lynn, and it might come back to Sindra then, and then it will come back once Sindra and Lynn have finished up. Then it will come back to myself, because I want to just introduce the practical applications of all this data science, and then we're going to have a demo from my colleague Guyana. So what I'm going to do now is I'm going to stop sharing, and I'll start reading the chat and any Q&As that come up, and I'll start responding. Sindra will probably come online. He'll unmute, share his screen, share his camera, and I will stop sharing. And then, Sindra, when you're ready to hand over to Lynn or myself, just let me know, okay?
I will. Okay, cool. Hey, everyone. Hopefully you can see my screen depicting the Julie landing page.
We see it, Sindra.
Great stuff. So I'll just take you through from start to the end, looking at discrete data in this instance. So this is our standard login page. I'll sign in, and then I'll have all my clusters. So clusters are the higher order of organizing, meaning like the top directory of data. Okay, I can see I have lots of clusters. I have lots of data. Luckily, they're all searchable, so I know what I want to see. I want to look at some IQC, incoming quality control data, which I have here. I'll click into that. Inside here, I have a bunch of different sensors, and I have some carbon sensors, etc. I'm going to click back because I specially made a tutorial cluster. So keep into the tutorial, and inside here I have 2 projects, which is sort of the next level, the lower level of data organizing. Another folder. Click into webinar, and here I have my data entries, which we call reports. And that typically is the reporting from one measurement instance, one session in the lab, or one parameter studied. So I will click into this report number 2, and we'll have a view of the data. So this is built up like a standard lab report with the operator who have conducted the experiments, the date, some obligatory fields such as a hypothesis, experimental protocol, that sort of thing, just to encourage people to put as much detail as we can, because as academics, we know that we need a lot of detail in order to reproduce, and also our memory as humans aren't that great. So if, let's say, people come in and go out of the organization, then we will have a good idea anyway, because we recorded all of this. So I'll just blow up these images a little bit for you so you can see what they actually are. So here we have some sensors that we have tested. And in this particular experiment, we want to test for the reversibility of these electrodes. And there's a table that I've taken here from electrochemical methods, fundamentals and applications. These are kind of checklists for whether your data is reversible or not, such as peak distance, 59 millivolts at room temperature, and peak current proportional to the scan rate, or the square root of scan rate, rather. So we'll look at some data. I'll pull it up. I've uploaded some data here, and this data is from CH instruments. So we do support a lot of different data types, and we'll be happy to support your formats as well. If I wonder what is supported, I'll simply just click to this contextual help menu, hover over, and then that will give me a list of supported file types. So you have some media types like images, and then you have some raw data file types that you can just drag and drop, or you can browse your computer. So we have some different files. I happen to know that this is voltammetry data, so I will go ahead and click discrete studies and add a study. We say it is discrete because the conditions are the same during the course of one measurement. Whereas if I would be changing, let's say, concentration or something like that during a measurement, it would be a continuous study. So that's the distinction. All right. So here we go. I'm loading up these data now, and they should pop up like graphs of our raw data. So let's see what we have. Okay, so here we have the raw data. They are recorded with different scan rates. That's one of the tests that you saw in this sheet, the scan rate dependency. Now we want to check what kind of dependency does that have. So I will click into my study and define some parameters that I check for. I will study specifically the height of the peaks. I'll just specify roughly where I'll find my peaks. You know, yeah, 0.25 volts, for example, and then add a window, so margins to where do we search for the highest point. Now, notice that this particular set of data, we start from the right. So we go from a high potential down to a low potential and up again. So actually this here is the reverse peaks. I'll have to check that as well. Let's see what that looks like. Cool. So now I have identified all the peaks. This is the window where I looked for them. And notice we have gotten a new card. The card is the container of these tables and plots. We have a plot first and foremost where we are mapping the current as the signal versus the scan rate. So you can see there's sort of a trend going on. This is the linear regression, and we have an R² of 0.93. Now the condition for this was actually square root of the scan rate. So actually, let's go ahead and change our regression method to a square root to see if we can get a better fit. And there you go. That's actually quite a lot better fit. We have an R² of 0.999, which tells us that the condition of the square root of the scan rate being proportional to the height of this peak is has a pretty good correlation. So this here could, in this particular experiment, we were looking at the parameter of scan rates. Most of the time when making sensors, we are looking at chemical concentrations. So if you imagine these curves to be a function of an increase in chemical concentrations, then that is what your model would output. So that is a brief look into how to handle discrete data in Julie. This does also work with other techniques such as amperometry, potentiometry, impedimetric data, which we will look at a little bit later, and then you can also specify your own. So it doesn't necessarily have to be electrochemical data. If you had other types of data, such as optical spectroscopy or temperature or whatever, that would also find its place here. For Julie, this is just numbers. Units are arbitrary. So I'll hand off to Lynn now for looking at the continuous case.
Sure. Yeah, thank you, Sindre. So I will take over and go ahead with the continuous measurement. So one really important technique for electrochemistry analysis is the chronal amperometry. Is important for the application for oxygen detection as well as for the glucose detection, as mentioned by Martin. So today I'm going to share a report that we use our real samples that we use for like one FDA application. And the purpose for this work is to compare our sensor being functionalized with the old enzyme and compare that with the new enzyme. So to populate the repository, we fill in all the experimental results report inside here. And to upload the file is the same way as what Xingzhen mentioned, that you can drop the file here or click the add button to upload your file. You can do multiple files or like a single file. So once you upload your file, you can check like your information by clicking the process. And on this data here under the curve and group, you can manipulate the data by giving it more meaningful description or put them into different groups. The next step you would need to do is to tell the software what you are running, because with this software we are able to run different techniques, including chronoamperometry and the one showed by Xingzhen, like cyclophotometry. And when you upload multiple files, you need to tell it specifically you only want to see the chronoamperometry. After you selecting the continuous study, and if you see here, there's a box to tell you the amperometry or like other technique. So in this particular case, we are using amperometry. This tab will appear accordingly after you're selecting the amperometry. So in this tab, under this tab, you have multiple selections. So what you can do quite a few things. Like for example, if you did multiple scans, you can just selecting the status for the scan. You can choose the first scan or the second scan. In terms of the axis, depending on like how you want to view your data, you can give it like different units, or like to do the log on the data. And some data, when they come in, you would notice that like not all the sensors, they are on the ideal case that you will have a perfect baseline, so that you have an easy comparison from one data to another. So what we will need to do is to set a baseline. So I just give a quick look for everyone for how the data looks like. So this is the raw data. As I mentioned, that like you can see the baseline, they are not all drawn down to 0, or they are not even like together. So with this, it can be due to capacitance or like some impurity on the sensor. It can be due to the polymer as well. Doesn't matter what is the root cause. If you want to have like an easy comparison by eyes, we will need to bring the data down to 0, so that like this 0 concentration can be easily viewed. This experiment was done by, we wait the sensor to be stabilized at 0 concentration, and then we made a first spike at 300 seconds. And we wait for around 100 seconds, and then we go to the second spiking. So every spike we did around 2 millimolar. So depending on your experiment, you could design like what is the gap should be. So I will go back to the toolbox to explain how we can do the step fit, because I mentioned earlier that like we are spiking the glucose into the solution at a stepwise manner. So what we need to do here is there are like some analysis buttons. You need to show extracted value, and you need to, if you are interested to see the statistical analysis on the data set we are collected, you will need to click this one. And also for individual sensors, because it not only like the batch information is important, but individual sensor analysis can be important as well. So after you click all those buttons, you will need to go to configure the event. So the way to configure the event is done by telling the software where is your 0 concentration. This is being selected, but I just remove one, and then to show you how we can't select. Oh, okay, just I delete one, which is the middle one and also the first one. So I just delete them to show you how we can add a concentration at each step. So if you see the green button here, you can just click in and tell what time you want to evaluate. For example, I know that like maybe the best time for me is near the position I spike in the new solution. So I just put 250. And the concentration, as I mentioned earlier, is 0 millimolar. So I put 0 here, and then I do the same for the second step. So I click the green button one more time, and I know my second step is around 350 because I don't want to step in the middle of any concentration, and then I know it's 2 millimolar. The other thing we can also do is that we maybe the data was collected by averaging the window as shown here. But if you feel like we are stepping in the middle, so you can change that to 25. And also, if you think, oh, this is not quite in the right place, so you can drag it in the graph. And once everything is done, you just click down and close this window. Everything will be analyzed for you automatically. So you can see this is the process data, which the baseline was dragged down to 0, and each step is defined by the window I gave to the software. And so like those are the process data and then original data. So the next thing is really important, especially if you are doing the medical device, is the reported information to tell you how the concentration changes with how the current is changing with the concentration. And also is telling you what you did to the data so that you can achieve this graph. Is really quite a comprehensive report on this whole page. As I mentioned earlier, that like I'm normally interested in doing the independent statistical evaluation on each sensor. So I have a LOD, LQ, and the intercept, which is a C0, and the slope analyzed in the full table here for all the sensor, and also it has an overall data. And also like there are some government body ask for the more detailed analysis. So we also give like how each concentration vary from the other. And also this represent your sensor to sensor reproducibility, which is the key factors for the report. The last one is reflecting the plot as shown above. So it's giving us how the concentration are evaluated at each sensor and at each concentration. So if you want to download the report, you just need to click these 3 dots, and it has a download raw data button, or you can download the raw data, or you can download the analysis results. But if you want to download a report, you just export it as a PDF. Once you did it, it gave you an option to show the signature. So like if you need to file this report, you can just put a signature there. So this is overall, this is everything on the continuous data. So I will pass this presentation to Sindre.
Okay, thank you, Lynn.
Thank you.
All right. So resharing my screen. And yet again, we're seeing some voltammetry data. Now in this context, I want to talk a little bit about how to treat lots of data in a fairly standard way so we can save time. I have some data here, and this is something we typically do when we're doing manufacturing runs. We have our electrodes. This can be screen printed electrodes. This can be wires, you know, but they can be laid out in a format such that you can coordinate to these sensors using space coordinates. So here we have a 2-dimensional layout, and what we can take samples from this sheet or this wafer in order to get an indication of what is going on and how does the reproducibility look like on this sheet by taking, let's say, samples that we think represent the entire sheet well. So in this case, we've done our standard voltammetry. Now I have lots of analyses I want to do this, and I have some criteria for certain features that I require in order to pass this sheet. Now, luckily, I've stored all of those in a template. So actually, I'm just going to go into templates, choose this template. Are you sure you want to apply this to your data? Yes, I'm sure. Yes, I'm sure. And here we go. Now it's selected my pre-submitted or pre-registered settings, let's say, that I use for all these tests. So I don't have to go in and click all the knobs and buttons every time. This applies also for the analysis that Lynn did. You don't have to go and specify all these time series every time if your data is looking similar. So this becomes specific, especially handy when you're working with a maturing product where the tests that you perform are standard, and you only want to perform the same analysis over and over and over again. So this is the real time saver. I'll take you over what I've done here just by clicking into the settings. So we did this before. We selected a region from which to extract the peak height. Now what's new here, I've added something called spatial mapping. So this is where we map this parameter into those space coordinates that we were discussing. I've also applied a feature check. So these are specific features which are marked by the purple regions where I extract certain parameters. In this case, I've chosen the anodic and the cathodic peak of this voltammogram, and I'm looking at specifically the difference in the x direction. That means the voltage separation, and the difference in the y direction. In this case, that would be the current difference between these two peaks. And I have set certain criteria that Martin also mentioned earlier. I want this to be less than this. I want this to be greater than this. You can also add other stuff, such as, I want the standard deviation to be between these two numbers, or any kind of parameters you choose. Okay, so I didn't do anything to the data now, so it doesn't update because we already did it. So let's go through this in order. I have my graph. I can zoom in and check that everything looks okay. I can scale it. And then I look at the overall statistics. So we have an average current of 5.2 microamps. We have a standard deviation of 0.29, which results in a relative standard deviation of 5.5% across this selection. Now this was 9 samples. This is just for giving you a little bit of a glimpse, but this n might as well be 100 or 200 or whatever arbitrary number you choose. This is the output from the wafer mapping. So this is a bundle of plots where we look at the space coordinate and the value recorded for the electrodes occupying that coordinate. So we can see from the color scale that we have blue to red from 4.8 to 5.6. So that means blue spots are lower than the average, which is around 5.2. And then red spots are then higher. So you can see now, if I took more samples here, I could, for example, identify hotspots that would be indicative of something going wrong with the production in that spot. Now, in this case, we're pretty okay. So the extremes are not deviating that much from the average value. If you look out to the sides, here we have the rows summed up. So the average plus an error bar, which represents the standard deviation. So you can see on this row, we're a little bit higher. It's around 5.5 as compared to the lower portion of this sheet, where we are around 5, you know. So that could indicate that we have a little bit of a trend. Maybe the electrode area is greater, for example, on the top. Maybe we have a thicker print. You know, maybe there's some kind of systematic event happening during our print session that we can address. And this here is the overall distribution plot. Lastly, I'll just show you, which Martin has already showed you as well, is that if we have these two peaks and we look at specific features, we set our criteria, then you can see that the criteria value in all these cases are fulfilling the condition, so we have a pass, which means the overall verdict for this sheet is a pass. And then similarly for the current, okay? Now, if I didn't fulfill, these would show up as red fails. So that would notify that there's something going on. We need to take a closer look. And if I wanted Martin then to vouch for this, I would simply add him as a reviewer. Martin Peacock. See if I remember his email.
No, it's all one word.
It's all one word?
Yeah,
Right.
no.
See, that's wrong. Yeah, there we go. Okay, you are now a reviewer of this report, so you have to approve this, Martin. So that is the summary of how to deal with data in production and manufacturing. You can see I got a little icon here, which indicates that I've shared.
Brilliant, Sindre. What I'm thinking about a little bit, Sindre, is that because of time, that I might do a quick introduction to Sense It All, and then let Guyana do it. So I'll let you kind of go off mic and go off camera, and I'll take over this screen sharing. I'm just going to do a very quick talk for about a minute, and then I'm going to hand over to a colleague called Guyana. So let me do that now. We're talking about, I mean, you know, very, if you look at what Lynn and if you look at what Lynn and Sindre were talking about, Lynn was sort of doing linear regression and Sindre was doing statistical analysis on manufacturing. But these are not just words for us, you know, we really are doing these things. But what's really making it powerful, to be frank with you, is the fact that we've automated all of this, you know, otherwise it would be, you know, and it's just also process as well that you saw. Sindre and Lynn in particular said, well, you know, we added the concentration of glucose every time at the same time. So there's a process in place which got implicated in the lab. They uploaded the data. Then literally Lynn can then write a template which will just analyze the data. And it's the same for Sindre as well. You need a process in place. Everyone buys into the process. They upload the data. And then we can just run our template. Now I'm going to change slightly gears and just sort of say, and all of this we end up really helps us implement it into products. So we do have a platform called Sense It All. Now at ZP, I know we're synonymous with electrochemistry, but Sindre said it as well that, you know, we're actually expanding out of just being electrochemistry. And he said, oh, well, we can bring all sorts of data into this, because in the end, most sensor data is a string of numbers versus, you know, maybe a time or something like that. So we are actually working with spectrochemical these days, and we're also working with electrochemical as well. And this is our way of sort of taking all this data science and all this lab work and actually turning them into minimally viable products really quite quickly. So when we sort of show this stuff on our website, we're always trying to build to the idea that people are actually going to make products. And, you know, so we've built this kind of platform called Sense It All. It can take a sensor, or we can take a sample, like a liquid sample if it's optical. You will see this in a minute. There's an app on the App Store called Sense It All, and Sense It All actually talks to Julie. So so far we've talked about lab data making its way up to Julie, where we then do, you know, data analysis on that. But actually we can also then take, let's say, field data. It could be field data coming from a hospital, field data coming from the environment, and that also makes its way up to Julie as well. And that's really great, see, because when we can process the data in Julie and send the data back to somebody's smart device, and that's something that we really kind of, work with a lot of startups, and this is kind of our way of turning what's otherwise just lab bound quite rapidly into something that is actually sort of customer facing. So if you see some links, by the way, don't worry about it too much, because we're recording this. If you registered for this and you're in the room at the moment, it means we've got your email, and I'll send you an email at the end where we'll actually definitely send you the video as well. So I say here, we're going to do a quick demo. So what I'll do is I'll turn off my screen. My colleague Anna will turn on her cameras and start sharing her screen, and then we'll have a quick demo from Ghana.
Yeah, hello everyone. I'm Ghana. I'm an engineer here in Simran Peacock, and I'll start with a demonstration of our SenseTalk platform. So first of all, I have a device with me, which you see it's fits inside my palm. It's pretty small, and it's designed to keep it not only in the lab but in the room environment too. So I have a couple of QR codes and sensors. I have a chili sensor and a ginger sensor. So today we will test two samples. One is the powder mixed with the chili pot puffer, and another one is a ginger shot mixed with the ginger pot puffer. So I'll just start with the demonstration straight away. So I'll put the chili cartridge inside the device. And then all I need to do is to click this up to connect the sensor with the pins that are on the top part of the device. So I will get a connection. The device is already connected. I press the button for 2 seconds and I see the green light, which means it's connected. And now I'll share the app. So we have the app available in both iOS and Android versions. Right now I have Android phone with me, so I'll just demonstrate with that. And I already logged in using my Julie credentials. It means the data that we will get today, it will be uploaded to my Julie account. So I'll just scan the QR code. Yeah. Just disconnect the device to show that later. So yeah, so the app is set up for the chili sensor right now because I scanned the chili QR code, and we can see that it says chili sensor, it says the sensor part number, it says the sensor batch number, and also it says assay steps, meaning how many steps it will take us to make the complete measurement. Now I need to connect the device. I'll just connect the Bluetooth, click the Bluetooth icon, and then I will see all available devices. Now I see the SenseTalk 1003 is available, so I will click on it to connect. And this will take a couple of seconds, and we see the device, we see the serial number of the device, and also we see the MAC address of the device. The next thing that I will do, I'll go to test settings, and I will write the sample ID. I'll just write chili test 1, and also cluster name. This cluster name will be the one that will be shown in the Julie. I'll just write it demo, and operator ID is just my name. So we will remember these two names, demo and Julie test 1, and then later I will show how to find them in the Julie. I'll go back, and then I click go to assay. Now the app shows the instructions and tells what to do. The first thing that I need to do is to select dilution factor. So this solution that was prepared yesterday is chili powder mixed with a buffer with 1:100 ratio. So I will choose 100 from the list. Then it says place and lock the cartridge, which I already did. Now I need to apply sample to the sensor. I will apply 50 microliter. So I'll just do it somewhere. Yeah. And then I will make sure that I'm covering all the electrodes, and also that I'm not scratching the electrodes with the tip of my pipette. now it's done. Now I'm clicking the Start Measurement button. So the measurement takes around 30 seconds. What it does, it runs the cyclic voltammetry, and the device sends the data to the mobile app, and the app sends it to the Julie. And the data is getting analyzed in Julie. And after that, we will see the results on the screen. And this whole process takes approximately like 30 seconds, and the device will be back with the results soon. Yeah, we see that it came back with the molarity and ppm and SHU values, which is COVID hit units. And we also see that upload was complete, meaning that we can find the data in Julie. I'll remove the cartridge, and I also show that the device LED bar turned green, meaning that it's the measurement was successful, and we can see the data. I'll go to Julie now, and you see my screen? Yeah, it seems like this.
Yeah, it's perfect.
Yeah. I'm already in demo cluster, but I can go back to show that it's there. We'll search demo, and I see it here. And also I will find the MAC address of my device, which is this one, I believe. And I see the latest one, Julie test 1. This is the test ID that I specified in the beginning of the test. I will click, and then we will see the results. We will see this cyclic voltammetry, and we will see the peaks, and also we'll see how the Julie analyzed the data. Now I will go back and I will click done, and we will see that device LED bar turned white, meaning that it's ready for the next measurement. I will click off the scan icon, and I will scan next QR code, and we see that it set up the device to run for the ginger sensor. Yeah, I'll just change the test settings saying that this is ginger. So we will find it in Julie with correct name, and the cluster name I won't change, so it will be again in the same cluster. Yeah, I'll move forward, and then I have ginger solution, which is 1 to 10 ratio, so I don't need to do anything. And I will place the ginger sensor with a cartridge, and then click it up so it's connected. Procedure is the same. Again, I will pipette 50 microliter of the solution. Yeah. And I will be careful to not touch the electrodes. If there are bubbles, I will try to get rid of them. Yeah, and it's ready. Let's start the measure. Yeah, and the process for this one is similar. So we are running the cyclic voltammetry and sending the device, sending the data to Julie, where it will get analyzed, and then we will see the results on the screen of the mobile app, and we can also find the results in Julie. And of course, we can do all the features that we have access to all the features that Sindre and Dean were talking before. So I'll just remove the sensor, and we see that it gets back with the results with molarity and also milligram per liter. And also it says the upload was completed. And when I click to Julie, when I go up, I need to refresh, and I see the ginger test 1. I will click to the report. Yeah, and here is the data. Yeah, I think that's it from my side, Martin.
All right, brilliant. Well, thanks, Gana. I appreciate you going through that. And I think our only reason for showing that in some ways is just to say, look, that what is happening and in that cloud is data science, that we're sending raw data to the cloud, and then we're actually using the Julie database to turn that raw signal into something that's meaningful for those end users. Those end users in this case are food scientists and food technologists, and as much as we all love electrochemistry, they're not so interested. Actually, they just want to know what's the pungency of this ginger, or what's the hotness of that chili in terms of capsaicin concentration. Now that platform that we're just showing, it's really what we call an OEM platform, an original equipment manufacturing platform. So if somebody has an idea and they want to get something tangible quite quickly, that's what that platform's actually intended to. So I appreciate the demo from Guyana, and you saw how quickly we could actually just reskin the app just using those kind of QR codes. I think something that I just want to emphasize is you noticed that we didn't do any kind of calibration when we did that run. And you also saw Sindra talk about, we use data science to determine the KPI, the key performance indicators of our screen printed electrodes, so that we know they're manufacturing them well. And then you saw Guyana actually benefit from that because she didn't need to calibrate in the field because the underlying manufacturing is so repeatable. So you can see that data science is both helping us in our manufacturing and helping us in our final application. And I know I said it already, but that's why we're getting away with it, because we're actually manufacturing these screen printed electrodes really well. They're all available for the R&D community, just as an FYI, but you can see we're the biggest users of our screen printed electrodes, and that's what really holds the quality up on this. So I think the agenda today, I wanted to give you a quick introduction as very high level. Sindra wanted to show you how to deal with discrete data and voltammetry, was it? Lynn wanted to show you how to deal with continuous time data, and that was glucose. We use glucose there. Sindra kind of emphasized, the fact that, the statistics that we're doing are not that complex, but it's the fact that we've automated it, which is really the value add. And, we do that for our manufacturing. And then we actually showed you that, we can automate this process of recording voltammetry, sending that voltammetry to the cloud, turning it into a meaningful number, be it parts per million for the ginger or being Scoville heat units for capsaicin, and then we were able to send it back to the app. So I think, thank you for coming today. No need to take notes. We've recorded this, and once we've processed the video, we will send you all a link to the video anyway. So maybe I just want to say a particular thank you to Aftab this afternoon from Technando, who has reached out to you all and sort of, asked you to come along. I appreciate you all coming and staying to the end. If you've got any questions, reach out to Aftab or reach out to ZP. I appreciate it's something like 6:40-ish maybe in India at the moment. So thank you very much. And thank you very much, Aftab.